So in this lesson we're going to learn what transcription factors are and how transcription factors regulate gene expression. We're going to also look at how RNA splicing results in different products from a single gene. And we're also going to look at how gene expression can be changed by epigenetic modification, including non-coding RNA, histone modification, and DNA methylation. And we're also going to look at in what ways epigenetic modification is important in ensuring cell differentiation. So, first of all, we've got to think about what gene expression is. Well, gene expression is when the DNA code of a gene is used to make a protein. Okay, the code is read, and during the process of protein synthesis, a protein product is produced. Now, all cells contain the entire genetic code for that particular organism or its genome. However, they only need to express the genes and make the proteins that the specific cell type requires. All cells need a number of basic housekeeping proteins for the normal cell processes to occur, but then the rest depends on what the cell's actual function is. For example, the beta cells in the pancreas, they make insulin, so the gene for insulin has to be switched on. Okay? But a lot of the other genes can be switched off, that it doesn't need. Okay? So the protein insulin will be expressed in that cell. Now, expressing a gene involves transcription and translation, the two parts of protein synthesis. It is a very energy expensive process, so there are many regulatory steps to ensure a protein is only expressed when it's actually needed. You can actually make different protein products from a single gene by modifying the messenger RNA or the protein product itself. By comparing the proteins found in different types of cells, scientists can actually measure the level of differentiation that has taken place and work out which genes have been expressed and which have been suppressed. So, what are transcription factors? Well, they are the most common way of controlling gene expression, and this is by switching on and off the transcription of certain genes. Transcription is when the DNA is unwound and zipped, and the messenger RNA is produced, which is complementary to the DNA code. Okay? It's the first part of expressing a gene and making a protein. Transcription factors are proteins that bind to the DNA in the nucleus and affect that process of transcribing the genetic material. Transcription factors, they bind to a particular region called the promoter region of the DNA, which is found just upstream of the gene. Now, some transcription factors bind to regions known as enhancer sequences. Now, these sequences can be thousands of base pairs from the actual gene in this case. And when the transcription factors bind to these enhancer sequences, they can actually change the whole structure of chromatin. Remember, chromatin is the wound up DNA in the nucleus. It's all jumbled and wound up. And when these transcription factors bind, they may make it less available by folding over certain areas of chromatin to the RNA polymerase and therefore controlling gene expression. The polymerase can't get in there to do transcription in the same way, or it might actually make it more available by opening up sections of chromatin for the RNA polymerase to get in there. Several transcription factors can be involved in the expression of a single gene, or one transcription factor may control the activity of lots of genes. A transcription factor could stimulate the expression of one gene and suppress the expression of another. And transcription factors are the most common way to regulate gene expression. So, what about RNA splicing? Well, this is another way to control the expression of a gene. So this, instead of actually controlling whether the RNA is produced in the first place or not, we're actually now going to change the RNA, okay? Make changes to that RNA code after transcription has occurred. Now, the gene is actually made of different sections called exons and introns. The exons are the important coding sections and the introns are the non-coding parts of the DNA. And when you make the messenger RNA in transcription, it is called pre-messenger RNA because it includes both these exons and introns. And the first step of RNA splicing is just to remove those introns. But actually, when you splice the RNA, you can actually sometimes remove some of the exons as well. Now, you can go further with this as well. An enzyme complex known as a spliceosome actually will produce the final mature messenger RNA. And in a process known as RNA splicing, it can join and rearrange those exons in lots of different ways. And this means that actually from one DNA code and one messenger RNA strand, you can end up producing a range of different proteins. So that old idea of one gene coding for one protein isn't really true. One gene may end up coding for lots of different protein products depending on how you splice the RNA. Okay? Now these post-transcriptional changes to messenger RNA lead to more variety in the phenotype. An example of this is in chicks. So as a result of the spliceosome activity, a single gene, one gene, actually produces 576 different proteins that affect the sensitivity of the hairs in the inner ears of chicks, allowing them to hear a range of frequencies. So, epigenetics. What is epigenetics? Well, it's a very exciting area of scientific research. Now, epigenetics is interested in studying genetic control by factors other than the DNA sequence. Okay, so we're looking at anything that can affect that gene expression that isn't specifically the DNA sequence itself. Okay, and in fact, RNA splicing is really an example of epigenetics. Now, the environment, your environment, could influence your cell biochemistry, and then that could alter the quantities of different epigenetic factors and therefore the development of specialized cells. Now, scientists are particularly interested in how epigenetic changes can be inherited by future generations. We're going to look at 3 intracellular systems that can interact to control genes. 3 examples of epigenetic factors. One is DNA methylation, another one is histone modification, and then we've got non-coding RNA. So, DNA methylation first of all. Now this silences a gene or a sequence of genes. What happens is that you get an addition of a methyl group, a CH3, by an enzyme called DNA methyltransferase when a cysteine occurs next to a guanine with the phosphate bond between them. So you add this CH3 group on at this point. And it's called a CPG site where this methyl group is added. And what that does is it changes the arrangement of the DNA molecule and it prevents transcription from taking place. Okay, so by methylating DNA, you can stop transcription from taking place. It blocks that process. DNA methylation has been found to be very important in embryonic development. Likewise, DNA demethylation is also important. This is when you remove a methyl group and you reactivate that DNA again so it can be transcribed. And actually scientists have found that demethylation of DNA may well be a key factor in the growth of tumours in certain cancers. What about histone modification? Now remember that eukaryotic DNA is wrapped around histone proteins to form chromatin in the nucleus. Now this allows it to be packaged in a nice orderly way to enable it to fit into the nucleus. How tightly packed it is affects the gene expression. Remember, RNA polymerase needs to get in there and access the gene. Now if it's all tightly packed together, it's harder for the RNA polymerase to get in there. So tightly packed chromatin, or heterochromatin as it's called, means less genes are going to be available for transcription. And likewise, the opposite of that, vice versa, loosely packed chromatin will allow more genes to be transcribed. So this is another example of an epigenetic factor, how tightly packed the histones are. Now histones may be modified by methylation, again adding a methyl group, or acetylation, okay, of specific areas called the histone tails. Now methylation causes nucleosomes to pack more tightly together, and this means genes are not going to be expressed as much, and acetylation does the opposite. It opens up the packaging of the nucleosomes, and transcription factors can then bind in there, and we're going to get more gene expression occurring. Now the last example of epigenetic factor is something called non-coding RNA, ncRNA. This is the fact that actually 98% of RNA that's produced from DNA is not actually used to directly code for proteins. It's actually involved in regulating transcription or modifying the product of transcription. So most of the RNA actually is involved in some kind of gene expression. Now RNA interference regulates gene expression during the development and in our defense against viruses as well. there's two examples to look at here: microRNA, which is formed from this non-coding RNA, which can then interfere and stop translation by targeting particular RNA strands, messenger RNA strands, and short interfering RNA, siRNA, which is actually the example where we can use it to defend against viruses, because this type of RNA can actually cleave messenger RNA and allow that RNA to be destroyed, so it's not expressed in that case. So non-coding RNA is another epigenetic factor that can influence gene expression. A little bit about cell differentiation, which is going to come up later on in the course in more detail as we learn about stem cells. But as an organism grows, the cells differentiate into lots of different specialized cells. Okay, they start off as a basic stem cell, but then can turn into numerous different types of cells. Now, most of the changes that cause the cell to differentiate into these different types are actually caused by these epigenetic factors, as we've been discussing. Now, these change what proteins are produced by the cell as it differentiates, and the change in epigenetic factors can be in response to internal stimuli within the cell or external factors in the environment for the cell itself. Now, how do we know so much about all of this stuff? Well, gene probes have been really helpful in helping us to understand how genes are expressed and which genes are expressed. Gene probes are basically small sequences that we use to bind to very particular sequences of DNA or RNA, and they're fluorescently labeled. So we pick the gene probe that we know is going to bind to that particular sequence, and then we can add it in there, and if it's fluorescently labeled, we can detect whether that gene has been expressed or is being expressed. Ideally, want to tag it to the RNA actually, because then you'll be able to see whether the messenger RNA is being produced for that gene and the protein will be manufactured.